I'm just going to launch into it to try and not run over time here. So many people here, if you've come to this meeting before or you've heard me talk before, have heard me proselytize about phylogenomics and the benefits of phylogenomics and why you should work on phylogenomics. You may know me through that. You may know me through my obsession with open access to publications. That's my license plate. If you want to talk about it sometime, I'd be happy to. Or you may know me from my blog now, I guess. And as my crack reporting, I was the first to reveal that Craig Venter was sequencing his own microbiome on his boat instead of sequencing the global ocean survey data. You should read my blog if you want to learn more about that. But what I'm going to talk to you about today is this Genomic encyclopedia idea, which is a project that I'm helping coordinate at the Joint Genome Institute. It is a massive undertaking with a lot of people involved, mostly from the Joint Genome Institute. And what I'm going to do is take you through sort of the history of this concept and idea and some background on why we think it's important to do. And so here's an overview of my talk. I'm going to give you just a brief overview of all the genomes that I think are, what I think about all the genomes that are out there and why there are gaps in those genomes. Not the gaps you all think about in terms of genome assembly, but gaps in which organisms are available. And then I'm going to give you an example of some of the initial attempts to fill in these gaps and why we think you should fill in those gaps, and then talk about this genomic encyclopedia idea. So we're all familiar now, we've seen lots of examples at this meeting, and everybody knows that there are now thousands of complete microbial genomes out there, and even more incomplete microbial genomes out there. And this includes all sorts of different types of organisms, the pathogens, the symbionts, well, we need more symbiont genomes, but otherwise, the extremophiles, all sorts of diverse physiologies, shapes, forms, functions, genome structures. The diversity is incredible. This has been done in a large part because of a lot of efforts by funding agencies and by research groups to obtain the genomes of diverse organisms. There are both these sort of coordinated top-down efforts to get genome sequence of major pathogens or of organisms of relevance to particular environments, like the Moore 175 project. There are a lot of diverse granting programs that have been available for individual researchers to get their genome sequence, and all of this put together has resulted in an incredible diversity of genomes that are available for people to look at, analyze, use, and study. And it does cover an enormous amount of ground. However, if we actually take a step back and we look at the tree of life in particular, in terms of the genomes that are available, the tree is not happy with what we've been doing in these genome sequencing projects. And the reason the tree is very angry at us right now is encapsulated in these slides that are basically taken from a paper that Phil Hugenholz wrote in 2002, pointing out the bias, taxonomic and phylogenetic bias in genome sequences that are available. So at the time, they were estimated to be about 40 phyla of bacteria. This is a phylogenetic tree based upon ribosomal RNA of those 40 lineages. Most of the genome sequences were from these three phyla, the proteobacteria, the firmicutes, and the actinobacteria. There were a few phyla that had a couple of genome sequences available, but they were very sparse, maybe one genome or 3 genomes available, but the rest of these phyla were no representative was available in the genome data for any of those phyla. And the same general trend is true for the archaea and, in fact, for the microbial eukaryotes. So we've done this amazing amount of sequencing, and yet there's this taxonomic gap in the available genomes that are out there. And the solution to this is to take a sort of top-down approach and look through the tree and identify gaps in the tree and sequence those genomes. Now, of course, this doesn't work very well with some of the ways genome projects are decided upon and funded, because there's no champion for that particular organism that just fills in a gap. It may not be studied by anybody. But we need those genomes to do a variety of things that I will tell you about in a minute. There are a lot of people out there who have recognized that a phylogenetic approach to sequencing genomes can be useful. This is what's done in a lot of the animal genome projects. So you hear about the first monotreme, the first jawless fish, the first shark, the first marsupial, the first of all these genomes, those are selected basically because of their position in the tree of life for animals. And the NHGRI has done efforts like this, and there are a lot of other projects that have taken this phylogenetic approach, but it hasn't really extended extensively in a top-down way to bacteria and archaea. There have been a lot of individual projects, so the DOE original sequencing that was done at Tiger, a lot of the genomes were selected by their phylogeny. There have been a lot of CSP projects from JGI, and what I'm going to tell you about is one sort of mini top-down project first that we did at Tiger that we're just finishing up, and then I'm going to tell you about what we found from that project, and this is what we call, it's a Tree of Life project. It was funded by the NSF Tree of Life program, and basically what we did is we took Phil Hugenholz's paper and a little bit of analyses that we did and went through the tree, and we identified, these are, again, the genomes that were available at the time were in these yellow groups. We identified lineages that had cultured representatives, but no genome available at the time. And we selected one representative from each of those phyla to sequence as part of this project. We did 8 total phyla. So this was going to open up the tree in terms of the phylogenetic diversity of genomes that are available. And what I'm going to do for the first few minutes here is to take you through why we think it's important to increase this taxonomic coverage. And why, I mean, this is not just us. Again, there are a lot of papers on this. There's a lot of work that's been done on animals and plants and fungi. And what I'm going to do is show you some examples for the bacteria, why we think it's useful to increase the taxonomic coverage. So for example, if you're interested in studying mechanisms of diversification, we've heard a lot of stuff here about lateral gene transfer, for example. You find some region of the genome that you're studying that seems anomalous, and you'd like to find out what type of organism the genes in that genome might have come from. So you compare it to the genomes that are available. If our genomes that are available represent a tiny subset of all the types of organisms on the planet, we're not going to be very able to place novel genes in one organism as to their donor or their relatives in other organisms. If you want to do gene discovery, so certainly you can go to novel environments like has been done by companies and by academic groups and look for novel types of functions. But if you sequence the genome of a new phyla, we've found consistently that even now, with lots of genome data out there, the genes in that genome appear to be relatively novel compared to everything that's characterized in other organisms. So if you want to have a higher probability of finding novel things, you can select genomes that are distantly related from everything else that's been sequenced. If you want to improve your ability to predict the functions of genes, there are lots of methods available that can be improved by having improved taxonomic coverage of genomes that are out there. One of them is a method that we like a lot, which is called phylogenetic profiling, where you look at the distribution patterns of genes across species and use that to help predict the functions of genes. And this is an example of a phylogenetic profile, which gets much better if you have a broader diversity of organisms to compare your genome to. And of course, this is the metagenomics session, and so I have to talk a little bit about metagenomics. We think that improving taxonomic coverage is going to be very beneficial to metagenomic studies for 2 reasons. One, it's going to help you bin metagenomic data to the type of organism that came from, and it's also going to help you do diversity studies with environmental samples. We've already done a little bit of this in the Venter Sargasso Sea paper. We did comparisons of a bunch of different genes and how what they tell you about the taxonomic groups that are present in the sample. One problem with doing this is that if you have, let's say, a REC-A gene from your environmental sample, and in a phylogenetic tree it groups with the cyanobacteria, you don't actually know if it's grouping deeply in the cyanobacterial group if it came from a cyanobacteria or another group that we just don't have REC-A from yet. So with ribosome RNA, we have such a good sampling that it works pretty well with ribosome RNA. But with every other gene that we'd like to use for environmental surveys, that taxonomic assignment, however you do it, if you use Megan or Normoblast or phylogenetic trees, it just doesn't work well if you don't have a big database to compare those genes to. The same is true for actual binning environmental sequences. So Nancy Moran talked about this glassy-winged sharpshooter dual symbiosis that we worked on with her. And one of the ways that when we took data from this mixed community of host and 2 symbionts in the sample, one of the ways that we used to bin the environmental sequences from that sample was by building phylogenetic trees. And for example, here's a sequence read that groups in a tree with Bacteroidetes genomes, which is where that second symbiont that Nancy talked about came from. If we did not have these Bacteroidetes genomes, we would have had no idea that this sequence came from that other symbiont. And so as we get more and more genomes from across the phylogenetic tree, we're going to be much better able to place metagenomic fragments and contigs into a context to improve your binning of environmental samples. And so final thing, so related to that, I could give lots of examples. We've taken those tree of life organisms, these 8 genomes, and gone to environments that we think they might be helpful for improving the binning. For example, Yellowstone mats, we took the data from John Heidelberg's project that he talked about yesterday and did evolutionary trees of all of those sequences. And when we add our new genomes, many of which are thermophiles isolated from hot spring environments, we are much better able to assign those environmental sequences to taxonomic groups. And so the last thing, I mean, I could list 100 of these. The last thing that I want to give as an example for why improving taxonomic coverage is useful is because it allows you to better infer the phylogeny of the organisms and relatives of theirs that are under study. And so I'm going to give you one example of doing this with genome studies, which is a study by Jonathan Badger, Naomi Ward, and me, Jonathan's here in the audience, on a genome of an organism called Hyphomonas, which in the 16S ribosomal RNA phylogeny and the classification of Hyphomonas, it was assigned to the Rhodobacteriales group. And if you look at the whole genome and even the 23S ribosomal RNA, it definitely appears to be in a different group. So if you concatenate all the ribosomal proteins in this organism, or if you analyze the 23S ribosomal RNA, you would assign it to the Collobacteriales group. So ribosomal RNA is great. It's a wonderful marker. It is not perfect for doing phylogenetic assignments of all organisms. And so by adding genome data, we're better able to place organisms into a tree of life, which can be very beneficial. So this is a tree of all the organisms, representatives of each phylum of organisms, including in red, are tree of life organisms. And when we add these organisms to the tree of life, based upon complete genome sequences, we are going to propose rearranging some of the phylogenetic groups that are out there, demoting some phyla, possibly suggesting other phyla. It doesn't change the whole picture of the tree of life, but it improves the resolution, in particular, of the deep branches in the tree. Of course, there's got to be another problem with this. Why do we need the Genomic Encyclopedia? Well, the tree of life still is not happy with us. And that's basically because when we did this tree of life project, we didn't have a lot of genomes available, so we thought one genome from each of these phyla is going to revolutionize our ability to characterize those groups. And a bacteriophylla is like 1.5 billion years old or more. The diversity within a phyla is just massive. And so one genome as a representative of each of these phyla is simply insufficient to do really any of the stuff that we think the benefits of taxonomic coverage is going to be for. So we think you still need to go through each of the taxonomic groups and fill in the gaps in the tree to really allow for these things that will come from increased taxonomic coverage, those 5 things, as well as a lot of other benefits from increased taxonomic coverage. So to do this, we need a more organized, systematic, top-down effort to get genomes from across the tree of life. And so that's where this idea of a genomic encyclopedia comes in. And this has been proposed, something akin to this has been proposed by multiple NAS reports, American Academy of Microbiology reports. There's a group primarily at Argonne National Lab that has proposed sequencing everything in Berge's manual, for example. There's a definite need for these sort of top-down approaches to sequencing microbial genomes. And that's what we're calling this genomic encyclopedia of bacteria and archaea. And to see the benefits of this and to test how we might go about doing this, Eddie Rubin at JGI has agreed to use JGI funds to do a pilot project for this. And so this is the pilot project. We're going to select, we have selected, genomes to sequence from across the tree. The goal is to finish 100 in this year, selected from across the tree, that is shotgun and close them. We might not finish closure in the first year. And then to use that to evaluate the actual utility of this tree-based sequencing. And the approach that we've taken is to select more than 100 genomes, about 200, to collaborate with culture collections, because the rate-limiting step in doing this sequencing is getting the organisms grown and getting DNA from them. and then to sequence 100 of those for which the DNA quality control appears to be good, and then to assess the tree-based sequencing based on that. So there's, as I suggested at the beginning, there's a large number of people, primarily at JGI, that are involved in this project. Project management for coordinating with the genome, the culture collections. The methods of sequencing are changing, so we need to coordinate with that. With Paul Richardson, the annotation and database needs, the selection of strains. There's a huge number of people involved, and I'm sort of representing this group as the coordinator of this effort. So I'm going to give you just briefly a little more detail on this selection process and our progress in this project. So how do we actually select the organisms to do for sequencing? We have chosen to focus initially on pure isolates, on cultured organisms that have been isolated and are in culture collections. And so what we first did is we merged the list of available genomes. So we did this from the Genomes Online database, which is a relatively up-to-date collection of all the available or in-progress microbial genomes. And we took the ribosome RNA tree of life from the Green Genes database, and we merged these, basically. We overlaid all the available genomes onto the ribosome RNA tree, and then we looked for gaps in the ribosomal RNA tree where there was a lineage that had no genomes in progress or available. And then we selected within those lineages organisms that might be of preference to the Department of Energy based upon their known biological properties. We focused on type strains, so in some of these lineages, there were no cultured organisms. We didn't do those, at least at the beginning of this project. We focused on type strains, and we focused on those that are available in culture collections, because that's where we think we can get a pipeline for getting the DNA. And we've been talking to a bunch of culture collections. ATCC is providing DNA for a couple of the organisms for this project for a fee, but DSMZ, which is a German culture collection, the national culture collection in Germany, has volunteered to do this for free. Given that I have a budget of $0 for this project, outside of the sequencing lanes, that was very convenient. And so we've been teaming up with DSMZ to provide the DNA for these projects. And again, we're working with other culture collections to hopefully add them in. But if we want to do 1,000 genomes in 2 years, we need to partner with these places that can grow diverse organisms, that can provide the DNA in the same quality for every project in order to streamline the pipeline. This is just a little plug for DSMZ. They've been amazing in the course of this project with quality control of the DNA. They're doing all the ribosomal RNA characterization, the pulse field gels, the quantification of the DNA, and then sending the DNA to JGI along with all the documentation of the organisms and the DNA quality control. So this is the status of the project. We have 78 DNA samples that have been received at JGI, 53 are awaiting library construction, 25 are in some stage of progress, most of them in library construction. There are a few that are actually in or near the end of production sequencing, and there are 94 for which we're awaiting material. And you can find the list of these at the GOLD database. So Nikos Kirpidis has been updating the list of all the genomes that are even those we haven't started, that are candidates for this project. And so the long-term plan as part of this project, there's a big gap in what I've told you about, which is if you focus on cultured isolates, you're going to leave out a lot of the lineages of bacteria and archaea that have no cultured isolate. So our hope is to also sequence lineages of organisms that have no cultured representative, and that way we will really fill in the gaps in the tree of life. So how are we going to go about doing this for uncultured organisms? There are a lot of possibilities. One is to get people to get them into culture, as you've heard some examples here. You can use enrichment cultures, and then shotgun sequence the enrichment culture. You could use a low diversity ecosystem. You could use flow sorting, microbeads, microfluidic systems. We're sort of playing around single cell amplification. We're playing around with all of these in the hope that then we will really be able to sample the tree of life if this project gets extended in the long run. And so to get this to be really successful, sort of my mission and a lot of the other people involved in the project, we really want community input into this project and engage the community in the long-term goals of this project to try and see what are the benefits and the advantages and how we can get people involved in all the different aspects of this project, and to try and team up with some of these there are more American Academy of Microbiology reports that are going to be related to this, so to try and work with them. And so in the end, I'm hoping that this is what we get. A very happy tree of life where the lineages are better sampled. I'll end on that and just leave up the acknowledgement side, but to say in particular I want to thank JGI and DOE and Eddie Rubin, who have been allowing some of the sequencing lanes at JGI to be used for this project, this pilot project.